This method creates lipid bilayers that mimic an infected cell surface in order to study the HIV virological synapse formed with primary human CD4 T cells. First, label proteins of interest with fluorescent dyes. Then, create lipid bilayers and allow the cells to form synapses. Fix and stain the cells with fluorescent antibodies for visualization of signaling molecules. Then, using total internal reflection fluorescence, or TIRF microscopy, acquire images of signaling molecules within the synapse-forming cells. Analyze the TIRF images to delineate the localization of active signaling molecules with respect to the virological synapse structure. This experimental approach can indicate whether cellular proteins like the lymphocyte-specific protein kinase LCK are recruited to the HIV virological synapse.
This method can help address key questions in HIV virology and immunology, such as which cellular components are involved in the virological synapse, what is the role in synapse formation, and how it may affect the HIV transfer from cell to cell. This bilayer system provides an end-face view of the synapse, with advantage of visualizing the synapse in high spatial resolution in a single plane. The results clearly depict the synaptic structure. Forming the bilayers and assembling the flow cell is akin to an art form. Here we demonstrate bilayer preparation on the biooptic flow cell.